Good afternoon, everyone. This afternoon, we are going to learn about cell division. Cell division is a fundamental process in living organisms that allows for growth, development, and repair. The two main types of cell division are mitosis and meiosis. Let's look at cell division by the type of mitosis. Mitosis is a type of cell division that occurs in eukaryotic cells, and it is responsible for multicellular organisms' growth, development, and maintenance. Let's look at the importance of mitosis. Growth and development. Mitosis is essential for growth and development of organisms, enabling the reproduction of new cells to replace damaged or old cells. Tissue repair. Mitosis facilitates the regeneration and repair of tissues by replacing damaged or lost cells. For example, in the morning, when you wake up and before you apply Vaseline on your body, you will realize that there are some whitish stuff or cells. Those are cells that are worn out and they have been replaced by the process of mitosis. Asexual reproduction. In some organisms, mitosis is the primary method of reproduction, allowing for the production of genetically identical offspring. For example, in the amoeba. Let's look at the key stages of mitosis. The first stage is known as interphase, prophase, followed by metaphase, anaphase, and telophase. Let's look at the key stages of mitosis in details. Interphase. Interphase accounts for 90% of the cell cycle. During interphase, a nuclei envelope bounds the nucleus. Then two centrosomes are formed by replication of a single centrosome. Each centrosome has two centrioles in animal cells. Chromosomes duplicate during S phase and cannot be seen individually because they have not yet condensed and synthesizing necessary proteins. Interphase is also divided into three subphases. We have G1, that is gap 1. Then we have the S, that is the synthesis, and the G2, that is gap 2. Below is an image showing the centrioles, that are two pairs, plasma membrane, nuclei envelope, the nucleolus, and chromatin. And this image is portraying the interphase stage. Let's look at the second stage of mitosis, which is known as prophase. During prophase stage, the chromatin fibers become more tightly coiled, condensing into discrete chromosomes. Each duplicated chromosome appears as two identical sister chromatids joined together at their centromeres. The mitotic spindle begins to form. The nuclei disappear. The centrosomes move away from each other. As you can see in the diagram, below, you'll be able to see the mitotic spindle, the separating centrosomes, the nuclear envelope, the centrosome with the attached kinetochore. Then you'll also be able to see the condensed chromosomes. Let's look at metaphase. During metaphase, the centrosomes are now at the opposite poles of the cell. The chromosomes convene on the metaphase plate, which is an imaginary plane that is equidistant between the spindle's two poles. The chromosomes centromeres lie on the metaphase plate. For each chromosome, the kinetochores of the sister chromatids are attached to kinetochores microtubules coming from the opposite sides. As you can see in the diagram, you can see in the middle is the metaphase plate. We have the kinetochore microtubule. We have the centrosome and the spindle pole. We have the nuclei polar microtubule and astra microtubule. Let's look at anaphase. During the anaphase stage, the sister chromatids separate and move to the opposite poles of the cell. The spindle fibers shorten, pulling the chromatids towards the poles. As you can see in the image below, there is increasing separation of the spindle poles. You can see the astromicrotubule in the other side of the cell. Then you can see in the middle, you have the polar microtubule. Then below, down, you have the shortening kinetochore microtubule. Telophase. During telophase, the chromosomes reach the poles and decondense into chromatin. A nuclear envelope forms around each set of chromosomes, as depicted in the diagram below. Cytokinesis. During cytokinesis, the cytoplasm divides, resulting in two daughter cells. Each daughter cell contains a complete set of chromosomes, as shown in the image below. Let's look at how mitosis is regulated. That is regulation of mitosis. The cell cycle, including mitosis, is tightly regulated to ensure accurate replication and distribution of genetic material. Otherwise, if accurate replication and distribution of genetic material does not occur very well, then it means this will pose problems in the offspring cells. Checkpoints such as the G1, that is gap 1, and spindle checkpoints, these monitor DNA integrity and ensure proper chromosome alignment before progressing to the next stage. Let's look at meiosis. Meiosis is also another form of specialized cell division that occurs in sexually reproducing organisms. Meiosis is responsible for the production of gametes, that is the sex cells, with half the number of chromosomes as the parent cell. Let's look at the significance of meiosis. The first significance is genetic variation. Meiosis generates genetic diversity through independent assortment of chromosomes and the crossing over during prophase. So this variation contributes to adaptability and evolution of species. The second significance is sexual reproduction. Meiosis ensures the production of haploid gametes, in brackets, the sperm and the eggs, with half the number of chromosomes as the parent cell. When fertilization occurs, the fusion of gametes restores the diploid chromosome number in the zygote. What does it mean? It means that the female or the mother will contribute 23 chromosomes, then the father will also contribute 23 chromosomes. So when a zygote is formed, it means that the diploid number of 46 chromosomes is restored in the zygote. The image below depicts cell division by meiosis. You can look at the interface, you can see the chromosomes. Prophase, the spindle fibers are there, metaphase, anaphase. The sister chromatids remain attached, then the homologous chromosomes separate. Let's look at the key stages of meiosis. The first stage of meiosis is known as interphase. So similar to mitosis, meiosis is preceded by an interphase that includes growth, DNA replication, and protein synthesis. Because for mitosis to start during the interphase stage, it means that growth has to take place, the DNA also has to replicate, and also proteins have to be synthesized. The second stage of meiosis is meiosis 1. So meiosis has stages. It has been divided into two. There is meiosis 1 and meiosis 2. So let's look at meiosis 1, prophase 1. So in prophase 1, the chromatin condenses and the homologous chromosomes pair up and undergo crossing over result into genetic recombination. Followed by metaphase 2, followed by metaphase 1, During metaphase 1, the homologous chromosomes pairs align at the metaphase plate. The next stage which follows is anaphase 1. During anaphase 1, the homologous chromosomes separate and move to the opposite poles of the cell. Followed by telophase. During telophase 1, the chromosomes reach the poles and a nuclear envelope may form around each of the chromosomes. Cytokinesis follows. When cytokinesis follows, this results into haploid cells. Meiosis 2, prophase 2. During prophase 2, the chromosomes condense again, forming new spindle apparatus, followed by metaphase 2, where the chromosomes align at the metaphase plate, followed by anaphase 2. During anaphase 2, sister chromatids separate and move to the opposite sides. Telophase 2. During telophase 2, the chromosomes reach the poles and the nuclear envelope forms around each set. Cytokinesis follows, resulting into 4 haploid cells. Anaphase 2, the sister chromatids separate and move to the opposite poles. Then telophase 2, the chromosomes reach the poles and the nuclear envelopes form around each set. Cytokinesis follows, resulting into 4 haploid cells. Let's look at the similarities between meiosis and mitosis. Similarities and differences between meiosis and mitosis. Meiosis and mitosis are 2 types of cell division processes. Meiosis results into formation of haploid cells, whereas mitosis produces genetically identical diploid cells. Meiosis involves 2 rounds of division, that is meiosis 1 and meiosis 2, while mitosis consists of 1 division. Meiosis is involved in sexual reproduction, whereas that is in the formation of gametes, while mitosis is responsible for growth, tissue repair, and asexual reproduction in some organisms. Let's look at genetic disorders and meiosis. Errors at times may occur during meiosis, and these errors lead to chromosomal abnormalities and genetic disorders. Non-junction means failure of the chromosomes to separate properly during meiosis 1 or meiosis 2, and this can result in an incorrect number of chromosomes in gametes. Examples of genetic disorders caused by meiotic errors. Meiotic errors include the Down syndrome, that is trisomy 21, and Turner syndrome, that is monosomy X. Thank you for listening.